In this chapter on the vasculature, we'll be looking at the structure of blood vessels. We'll look at the structure of the different types of blood vessels and their major functions and the tissues that make them up. This very schematic picture lays out the basic idea of how the circulation works in the body. The left side of the heart is pumping oxygenated blood from the lungs out to the body, and the right side of the heart is pumping the deoxygenated blood back to the lungs. And so the color scheme that we see, red versus bluish purple, is just looking at the oxygenation state of the blood. The vessels that carry blood to the heart are veins, and the vessels that are carrying blood away from the heart are arteries. So the pulmonary artery and the systemic veins are all colored bluish purple, and the pulmonary veins and the systemic arteries are all colored red. Notice also here that there's a special arrangement for the capillaries that go through the digestive tract. Instead of going right back into the venous circulation on the returning side, that actually goes through a special vein called the hepatic portal vein to capillaries in the liver, where the liver can then process that blood, detoxify it to some degree. Otherwise, the circulation basically runs arteries to capillaries to veins, heart, arteries to capillaries to veins, heart, etc., etc. We're going to look at the specific structures of these different vessels in more detail. There are three major types of vessels that I just suggested before: arteries, veins, and capillaries. Arteries and veins have some structural commonalities between them. They're both made up of three layers of tissue referred to as tunics. The innermost layer is called the tunica intima. It's a simple squamous epithelium that's referred to as the endothelium, and its associated connective tissue. In arteries, there's additionally a layer of dense elastic connective tissue called the internal elastic membrane. The tunica media is a smooth muscle wall in both arteries and veins. In arteries, there's an additional layer of dense elastic connective tissue in the tunica media called the external elastic membrane. And then the outermost layer is the tunica externa, which is a connective tissue sheath around the blood vessel that helps anchor it in place within whatever organ that it's passing through. For the larger arteries and veins, there are in fact smaller arteries and veins called the vasa vasorum, providing nutrition to the outer tissues of those vessels. And there are, and not just for the larger vessels, there are nerves connecting to that, largely postganglionic sympathetic fibers called the nervi vasorum, which release norepinephrine to cause constriction of the blood vessel, or in special cases the ones that release acetylcholine to cause dilation of the blood vessel, or the parasympathetic system that just has acetylcholine releasing fibers there. This micrograph shows an artery and a vein side by side. Most often you'll find blood vessels that are going to and from the same place to be located right next to each other. This could very easily be the splenic artery and the splenic vein, bringing blood to and from the spleen. There's no way of knowing for certain that they are, but let's work with that idea. Now, notice that the wall of the artery is thicker than the wall of the vein. That's because the tunica media in an artery is thicker. There's more smooth muscle in the artery. Also notice that the artery is staying pretty much in a circular profile, even though there's no blood in it, while the vein has collapsed. That's because of the elastic arteries, the elastic tissue in the artery. It maintains its shape even when it doesn't have anything in it. The vein, however, stays open because of the blood that's in the vessel. If you can sort of mentally enlarge or inflate the vein, you'll also notice that it's larger than the artery. There's more internal diameter of the vein than of the artery. There's more pressure behind the blood in the artery than in the vein because the arteries are closer to the heart. There's a trade-off in diameter, which leads to less resistance in the vein to help keep the blood flow consistent. You need to have blood going into and out of an organ to be at the same rate. If not, then either the blood's going to accumulate in the organ or it's going to drain out. So the trade-off between pressure and resistance is evident in the fact that the artery is thinner and the vein is wider, diameter-wise. This picture demonstrates the finer differences in structure within three different types of arteries. There's the elastic artery, which has a preponderance of elastic tissue in it, a muscular artery, which has a greater amount of muscle tissue in it, and the arteriole, which is a microscopic blood vessel. These three pictures are not drawn to scale. It might seem like the elastic and muscular arteries are pretty close in size and structure, but really pay attention to the layer of elastic tissue in the tunica intima. And it's a much smaller drawn structure in the elastic artery than in the muscular artery, but it's actually a much more pronounced part of that artery. And also the muscular artery has a thicker tunica media for its size than in the elastic artery. Even though it looks like the elastic artery has a thicker tunica media here, keep in mind that they're not drawn to scale. And then finally, in the arteriole, you can really see the difference in scale because we can see the individual smooth muscle fibers in the arteriole much better than in the full-sized arteries. Now, I'm using the word arteriole and making a very concerted effort to pronounce the O in the ending there so that you can tell the difference when I'm saying arteriole versus arterial. The less pronounced ending is arterial, which is the adjective form of the word artery. And so if I'm talking about arterial flow or arterial circulation, that's what's happening in the arteries. If I'm going to talk about an arteriole, I'll make sure to really pronounce the O sound at the end so you can distinguish the words from each other. These are three capillaries. Capillaries are a different type of blood vessel compared to arteries and veins in that they're only made up of an endothelial layer and the associated basement membrane. Now, you can think of that endothelial layer as a tunica intima, but there aren't any other layers. There's not a tunica media, there's not a tunica externa. So it's not common to call it by that name, just the endothelium. Here we see a range of types of capillaries, really looking at leaky to rather solid-walled capillaries. The continuous capillary is one that has tight intracellular clefts so that not a whole lot can leak out between the cells. And then the cells themselves are pretty solid. Anything that's going to get across the cells is probably going to move through an active transport process like pinocytosis or endocytosis and exocytosis. The basement membrane is also a complete cover on the outside of the capillary. It's essentially a collagen fiber mesh. You could think of it as a loose connective tissue without any real cells to it. Then a slightly more leaky version of a capillary is the fenestrated capillary. The difference with the fenestrated capillary is that the cells that make up the wall of the endothelium have little gaps in them, little windows, which is what fenestration means. And small molecules can pass through that a bit easier. The fenestrated capillary still has a complete basement membrane surrounding it. And then when we get to the most leaky capillaries, we're talking about the sinusoidal capillaries. Those are going to have wide intercellular gaps. The fenestrations of the fenestrated capillary aren't really present, but that's not a big deal because the wall is so leaky. And then the basement membrane is incomplete. So there's actually a way for things to just leak right out of the blood and into the surrounding tissue. We see the sinusoidal type capillaries in organs that have a very rich blood supply, like the liver and the spleen. Fenestrated capillaries are found in most common capillary beds, moving towards a more continuous type found in organs where we need a very tight control over what's moving in and out of the blood. For example, the blood-brain barrier regulates what can pass from the blood into the nervous tissue of the central nervous system. And so that's going to have continuous capillaries in it. Here we see the difference in structures within veins. Now, it might look like from this picture that there's three different types of veins, but really large veins and medium-sized veins are pretty much the same thing. And what's different are the venules. Venules are microscopic vessels, like arterioles are on the artery side. Then we have the medium-sized and large-sized veins, which are really just the same thing of different sizes. In the medium-sized vein picture here, they demonstrate a valve. And a valve is going to keep blood moving in only one direction through the blood vessel, just like it does in the heart. They're may be valves in larger veins. I've not been able to find a reliable source to say one way or the other, even though this book suggests that there's not. But I don't have a reliable source saying that one way or the other. So there really are just two types of veins. The vein itself, which is macroscopic, you can see it with your naked eye, and venules, which are microscopic. Looking more closely at how the arterial and venous sides of circulation interact, especially across capillaries, we have this figure that gives us a little bit of an insight. Now, arterioles branch off a smaller vessel called a metarteriole, which gives rise to the capillary bed. That eventually joins into the venule side of circulation through what's called the thoroughfare channel, which is really the venous side of the capillary bed. Capillaries emerge from the metarteriole and eventually merge back into the thoroughfare channel. Here we see small smooth muscle sphincters wrapped around the beginning of each of those capillaries, which can pinch off, as is suggested in this picture, so that blood flow doesn't go into the capillary bed but stays in the medarterial thoroughfare channel. And that can shunt blood through the tissue if the blood isn't needed for that tissue in any great demand at the time. Another way that blood can be shunted away from capillary beds from particular regions is through an anastomosis. We see here an arteriovenous anastomosis, or AV anastomosis, meaning that it goes from the arterial side to the venous side. There's also arterial anastomoses, which is when a bypass grows around a blockage in an artery. We see these sorts of things especially in coronary arteries. Now, you've probably heard of bypass surgery. That's just an artificial way of doing the same thing. But there are actually arteries that can branch out new growth that go around a blockage to keep circulation going. Now, that's more common in older people. A 60-year-old person is more likely to form arterial anastomoses than a 40-year-old person, which goes to show why 40-year-old people suffering heart attacks usually have a worse time than 60-year-old people. Consider this question. When you feel that you've reached an answer, hit the next button to go into the next slide, which will reveal the correct answer. The fenestrated capillary is the capillary that has openings or fenestrations in the endothelium, but still has an intact basement membrane. It's probably the most common type of capillary, but I don't have any strong data to support that one way or the other. Now, choice D here, the lymphatic capillary, isn't something that we've talked about yet, but will come up at the end of this chapter. It's actually part of the circulatory system, just not the cardiovascular circulation. And it's important for recovering water that's lost at capillaries back into the blood.